Phosphorylation of receptor cytoplasmic tails by tyrosine kinases concentrates intracellular signaling molecules around the receptors. Phosphorylation of enzymes and other proteins by protein kinases is a common general mechanism by which cells regulate their biochemical activity and has many advantages as a control mechanism. It is rapid, not requiring new protein synthesis or protein degradation to change the biochemical activity of a cell. It can also be easily reversed by the action of protein phosphatases, which remove the phosphate group. Many enzymes become active when phosphorylated and inactive when dephosphorylated, or vice versa. The activity of many of the protein kinases involved in signaling is regulated in this way. Another and equally important outcome of protein phosphorylation is the creation of a binding site for other proteins. This does not alter the intrinsic activity of the phosphorylated molecule. In this case, phosphorylation is used as a tag, allowing the recruitment of other proteins that bind to the phosphorylated site. For example, many kinases involved in signaling are associated with the inner surface of the cell membrane and can act only and efficiently upon their target proteins when these are free in the cytosol. Receptor activation and the phosphorylation of membrane-associated proteins can, however, create binding sites for these target proteins. Cytosolic proteins that bind to phosphorylated sites at the membrane are thus concentrated near the kinase and can in turn be phosphorylated and activated. They can also, in some cases, be activated simply by binding to phosphotyrosine. This is an example of allosteric activation, as binding the phosphotyrosine leads to an alteration in their molecular conformation. Proteins can be phosphorylated on three classes of amino acid: on tyrosine, serine, threonine, or on histidine. Each of these requires a separate class of kinases to add phosphate groups. Only the first two are currently known to be relevant to signaling within the immune system. And that's phosphorylation of receptor cytoplasmic tails by tyrosine kinases concentrates intracellular signaling molecules around the receptors. Have a great day.